Hi Ninja Nerds, in this video we are going to talk about protein digestion and absorption. We have a lot of things to talk about, so let's go ahead and get started. Alright, so what we're going to do is we're going to take a scenario, just like if you guys haven't seen already, please go see our video on carbohydrate digestion and absorption. We did kind of the same diagram, and we're going to do the same diagram for lipids as well. So what we're going to do in this situation is we're going to start eating a nice chicken, like drumstick, alright? There's tons of packed full of proteins in there, right? So we're going to digest this drumstick. But here's what I want to do before we start getting into all the details of protein digestion and absorption. What I want to do is I just want to get this quick little basic overview, because there's going to be a lot of little stuff that we're going to talk about in this video. So I want us to get just a basic understanding of what this protein digestion is going to take, like what kind of events are going to take place and what kind of events will take place in the absorption as well. So when we talk about protein digestion, what I want you to remember here is in the basic terminology, when you talk about anatomy or biochemistry and stuff like that, is you hear the terms anabolism and catabolism. Well, protein digestion is primarily going to be a catabolic pathway. It's a catabolic pathway. Now, what that means is you're going to be breaking down large molecules into their small little individual units. What do I mean? Just like when we talked about in carbohydrate digestion, we're taking polysaccharides, like we took amylose and amylopectin, and broke them down into monosaccharides. In the same way, what we're going to do is we're going to take these large proteins, because all proteins is they are going to be these special macromolecules that are consisting of amino acids. So here I'm going to put all these dots here. If you guys have watched our videos, you've seen this probably once or twice before. This is a protein. This is what we're going to have. We're going to consider this to be a protein. Now proteins, we can say that they're like large polypeptides. So we can say here this is going to be our protein molecule, and in this case it's going to be a very, very large polypeptide. What we want to do throughout this process is we want to break down this protein into its constituent units. And we're going to do it through a mechanism which we call hydrolysis. It's going to be primarily through hydrolysis mechanisms. What does that mean? That means that you're going to be adding water into the reaction to break the different bonds. In this case, you're breaking peptide bonds. So it's going to be the hydrolysis of peptide bonds. And we'll talk about what peptide bonds we're actually breaking when we get into this different specific enzymes. But here's the basic thing. We're going to take this big protein unit, this large polypeptide, and break it down into each individual monomer. All right, and in this case, that would be these little things here, these little dots. And these little dots that we're going to break it down into is going to be your amino acids. So now, proteins are what we're going to refer to as a polymer, a long chain of these different monomers, or small little basic units, and we're going to break down this big polymer into these small little units called monomers. The reason why is it's really, really, really hard and inefficient to digest and actually absorb these different types of proteins. So because of that, we want to break them down into smaller little components because it's going to be more efficient and easier to absorb the amino acids in that way. So now that we got this basic concept that this protein digestion is a catabolic pathway breaking down proteins, which are large polypeptides, just components or constituents of amino acids, we're going to break them down by a hydrolytic mechanism, meaning we're adding water into these bonds. What kind of bonds? Peptide bonds. And we're going to break it into each individual monomer or basic unit, which is the amino acids. So now that we understand that, let's go ahead and get started and start digesting this chicken piece. Okay, so now we're going to start digesting this piece of chicken. So let's say first we come to a special organ that is a part of the GI tract. All right, this guy, we talked about this one in the gastric secretion phases, specifically in the cephalic and in the gastric. Well, there's special types of cells, right? There's special types of cells here that are called chief cells. And these chief cells, they were the ones that were primarily responsible. So we'll put here chief cells. All right, these are our chief cells. And what these chief cells are doing, if you remember, is they were secreting a protein digesting enzyme called pepsinogen into the lumen here. Well, what we're going to do is we're going to say that this is the lumen. This is going to be the GI tract or the alimentary canal here. So we're going to say here we're going to secrete this molecule, this pepsinogen, into this portion here of the GI tract. So we're going to say that we're at the level of the stomach because there is no protein digesting enzymes in the oral cavity, the pharynx, or in the esophagus. So the first organ that we come into contact with this protein digesting enzyme is in the stomach. There is another one called renin, but we're not going to talk about that because it's really only functional in younger kids to digest milk protein. So we won't talk too much about that. So now first thing I want to do is we're going to bring pepsinogen into the cell, into the lumen. When the pepsinogen is brought into the lumen, if you remember, we talked about there was these parietal cells. And the parietal cells were secreting a molecule called hydrochloric acid. And what the hydrochloric acid was doing is it was helping to be able to decrease the pH in the lumen of the stomach. And what that does is this pepsinogen is an acidic type of enzyme. It loves to be in acidic environments. So in acidic environments, around a pH of, we can say anywhere 1.8 to 3.5, this is its optimal range here, the hydrochloric acid concentration, if it's nice and high, will stimulate the conversion of pepsinogen into its active form, which is called pepsin. Sweet deal. Now, what does pepsin do? Pepsin is really cool because it's kind of like a auto-activator. Because you know what else is really cool about pepsin? Is any other pepsinogen that is secreted, this pepsin, the active form, so here's the active form of the enzyme, this is the inactive or the precursor form of it. This activated pepsin can come over and stimulate pepsinogen, more pepsinogen to convert into pepsin. So it's kind of like an auto-activator. Once he's active, he can go and activate more pepsinogen molecules and turn them into more pepsin. So that's a pretty cool thing too. It seems a very efficient mechanism. All right, cool. But again, as you know, if the pH is getting really, really high, anywhere above 3.5, it can convert the pepsin back into pepsinogen because this enzyme does not like to be in alkaline situations. It prefers acidic situations. Now here's our protein. We're going to use that diagram here. And again, if you remember, proteins have a amino end and they have a carboxy end. They also have a alpha carbon with a specific R group, but we're not going to talk about those in this video. Again, this isn't a biochemistry video. This is going to be specifically on the physiology of it. But here's going to be our protein, but we're going to refer to this as a very large polypeptide. So here's our large polypeptide. What we're going to do is we're going to bring this pepsin over here. Pepsin is so cool because he actually loves to break down large polypeptides. So what he does is he comes over here and he converts this large polypeptide into smaller polypeptides. So now what I'm going to do is I'm going to break this large polypeptide up. Let's say we break it into like 2 units. So now look, I'm going to have something like this. Here's an amino end. Here's a carboxy end. And here's going to be like a couple amino acids here, right? And I'm going to have another piece here. Let's say I have another piece just like this. And again, here's the amino end. And here is the carboxy end. And we'll have some amino acids on this thing as well. So now we have, from this large polypeptide, we have smaller polypeptides. So now what we did is we had the pepsin. Pepsin came in, cleaved some peptide bonds, and converted this large polypeptide into a small to multiple small polypeptides. You know what's so cool about this enzyme? It digests or breaks specific bonds. So pepsin is a pretty cool enzyme, and what pepsin does is, imagine here for a second, I'm going to take here a chain of like a peptide here, right? Here's our polypeptide, or large polypeptide. And let's say here I have a string of amino acids here, but there's some particular amino acids here that he really likes to target. You don't have to know their structure like completely. I just want you guys to get an idea here of what they are. They're aromatic amino acids. So he targets aromatic amino acids. One of them is called tyrosine. So this is called tyrosine right here. This is one type of amino acid that he likes to cleave like on the carboxy side. Another one is called phenylalanine. So another one is called phenylalanine. So this right here is called phenylalanine. And there's other ones. I'm not going to keep going through these, but here we'll have one more, and this is called tryptophan. So what he likes to do is he likes to break bonds on the carboxy side. So here's the amino terminus, here's the carboxy terminus. He loves to break the actual bonds on the carboxy side or carboxy terminus of amino acids such as tryptophan, phenylalanine, and tyrosine. By doing that, we're going to break these large polypeptides into smaller polypeptides. Cool deal. That covers this part. Now we come to the next segment. So then what happens is the stomach will actually undergo its motility. It'll undergo peristalsis where it'll contract and squeeze out some of the chyme into the first foot of the small intestine, referred to as the duodenum. In the duodenum, we have special types of mucosal cells that pick up the different concentrations of the chyme. Like if it's high in protons, if it's high in different types of carbohydrates, if it's high in protein digesting products or fats. And what it does is it signals the release of a bunch of different types of hormones. Some of these hormones are like cholecystokinin, secretin, GIP, which is glucose, insulinotropic peptide. There's a bunch of these different ones. But the one that's really, really significant in this is the cholecystokinin, CCK. What cholecystokinin does is he comes to the pancreas and he stimulates a special area in the pancreas called the acinus. So what the acinus is it's just kind of like this little bulbed out region here. So imagine I kind of like zoom in, like it's usually right here. What happens is this acinus has a duct that drains it. Like imagine there's the duct. And this is lined with what's called acinar cells. And what happens is CCK, or cholecystokinin, will act on these different cells here. Imagine there's a receptor for the cholecystokinin. What it'll do is it'll trigger these cells to start releasing different types of digestive enzymes. And the digestive enzymes that it releases are going to be proteases. There's a bunch of other ones like amylase and lipase and nuclease, but in this video we're only talking about the proteases. What are some of these proteases that are released into the duodenum, particularly through what's called the hepatopancreatic ampulla? It's just where the common bile duct and the main pancreatic duct fuse. They form what's called the ampulla of Vader or the hepatopancreatic ampulla. And then whenever there's a muscle there that relaxes, it opens up this substance, the pancreatic juice and even bile, to get released out here into the duodenum. So where we're at right now is we're saying we're at the level of the duodenum. Now, once we're at the duodenum, there's a bunch of different enzymes released here. Let's do these in this purple color. There's a bunch of them. We're going to talk about 3 of them. Okay, the first one that we're going to talk about is what's called trypsinogen. Now here's the thing, when I told you that we release these proteases, the proteases are technically inactive. They get activated in the lumen of the duodenum, so they're secreted in the inactive form. When they get into the duodenum, this trypsinogen molecule is so cool. There's a special enzyme that's present on the epithelial cells, the simple columnar cells of our duodenum. This enzyme right here is called enterokinase. 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 And what enterokinase is, it's kind of like a brush border enzyme actually. And what it does is whenever trypsinogen comes, it can get activated by that enterokinase enzyme. So the enterokinase can then stimulate trypsinogen. Well, basically it just cleaves off a specific peptide sequence off of this enzyme and turns it into the active form. The active form of this enzyme is called trypsin. So the active form of this enzyme is called trypsin. Other chemicals that are released into this vicinity that follow this is going to be another one, and this chemical is called chymotrypsinogen. Chymotrypsinogen. And there's two other ones. One is called pro-carboxypeptidase. And there's one more which is called pro-elastase. What happens is trypsin is so cool that once he is activated by the enterokinase enzyme, he can do 4 things. All right, ready? He can take, and you see this trypsinogen that was activated by enterokinase? We're going to have more of it, right? Because we're releasing out tons of these enzymes, thousands. This trypsinogen can get activated by the already activated trypsin, because trypsinogen got converted into enterokinase. There's going to be a ton more trypsinogen coming out here. When more of the trypsinogen come out here, this already activated trypsin can activate some of those trypsinogen molecules and convert those trypsinogen molecules into more trypsin. That's cool. Then this trypsin, we're going to take this one as example here, he can come and activate every single one of these enzymes. So what happens is chymotrypsinogen is going to be activated here. Let's do the arrows in different colors so we don't confuse it here. All right, so let's do this chymotrypsinogen in a red color. Let's do this one in this blue color. And then let's do this pro-elastase in this pink color. What trypsin will do is he will come and stimulate each one of these steps. If he stimulates each one of these enzymes, he converts them into their active form. Because again, under this is the inactive form. Inactive, same thing here, inactive, meaning they can't do anything. They can't cleave any type of polypeptide yet. So now what happens is trypsin is going to come down here and activate chymotrypsinogen. He'll cleave off a specific sequence there, turn him into the active form, which is called chymotrypsin. All right, sweet. So now we have chymotrypsin. The next thing is he'll cleave a portion of procarboxypeptidases and he'll cleave off a portion of proelastase, converting them into their active enzymes. So get rid of the pro on both of these. So you're going to have carboxypeptidase. And there is a type A and type B. We're not going to worry about that right now, but just do know that there is 2 types. And then it's going to convert proelastase into elastase. So now we have 4 pancreatic proteases that we want to talk about now. Because we had pepsin, and if you remember, pepsin broke down large polypeptides into small polypeptides by cleaving peptide bonds where there was tryptophan, phenylalanine, and even tyrosine on the carboxy side of it. These enzymes, they kind of target around the same area, particularly chymotrypsin. He'll target the same thing. So imagine here we have these small polypeptides. We're going to take it through here now. What happens is we'll take each enzyme one at a time. So let's take the first one, which is going to be trypsin. So let's take him first. The first thing is going to be trypsin digestion. If trypsin acts on this small polypeptide, he specifically cleaves bonds where there is a lot of lysine and arginine, okay? So he'll cleave a lot of peptide bonds, specifically where there's a lot of lysine and there's a lot of arginine. So that is what he is going to focus on. So wherever there's a lot of these 2 guys, he's going to cleave these guys here. So he'll cleave the peptide bonds, particularly on the carboxy side. So for example, pretend that amino acid right there is a lysine, Pretend that that amino acid right there is an arginine. What he'll do is he'll come over here and he'll break these bonds right there. Okay? So he'll break these bonds. There is like an exception, but again, this isn't a biochemistry lesson here, but just remember if you want to, he can't do it if proline is like right next to them. Okay? But again, not super specific here. Just know that trypsin can break down small polypeptides into even smaller polypeptides. Particularly, we would like if it can break it down into maybe something like this. This is called a tripeptide. So we have 3 amino acids together. This right here is called a tripeptide. Another one that we would like to see, ideally, is maybe even dipeptides. Okay. Then there's the next one. So this is going to be done by trypsin. So trypsin is primarily going to be targeting the bonds which are going to be having lysine and arginine there. All right? Then you go to the next guy, chymotrypsin. So let's take another small polypeptide here, and let's treat this one with chymotrypsin. So we'll have this one right here, because this guy and this guy have the same attacking point. So here's my small polypeptide here, and I am going to use in this one chymotrypsin. So now I'm going to use chymotrypsin. And when chymotrypsin acts here, he particularly targets the same thing as pepsin did. So if you remember, it targeted what amino acids? It targeted things like tryptophan, phenylalanine, and tyrosine. All right, we already had all these here. I might as well, just for kicks, let's just throw tryptophan in there. All right, tryptophan is kind of like this here, if you guys, I know you guys are interested, right? We have a 5-carbon ring right here. You have a nitrogen right here, right? And you have a benzene ring coming off here like this, right? And there's even one more bond like this right there, okay? So this right here is tryptophan. But remember, we said there was 2 different things here. If we cleave this bond, sorry, not this one here, and this one here, this was done by what type of enzyme? Well, it was done by pepsin. So this could be done by pepsin. Well, guess who else can come and do the same exact thing? Chymotrypsin. So chymotrypsin can also target the same exact thing there. Okay, so what type of things will he target here? We're going to denote them here. He'll target peptide bonds with tryptophan, TRP, peptide bonds with tyrosine, TYR, and peptide bonds with phenylalanine, and specifically on the carboxy terminus. When he does that, he's going to break down these small polypeptides into what? Same thing that trypsin did, dipeptides and tripeptides. So again, this will be our tripeptides, and this will be our dipeptides. Okay, cool. So that's what we know so far. One more thing is this procarboxypeptidase and this proelastase. So now what we're going to do is we're going to take these polypeptides here, these small ones, and let's keep them going here for these guys. Okay, so what carboxypeptidase and elastase do is they target in the same exact fashion. So pretend here I'm going to put carboxypeptidase and elastase. And what they're going to do is they are going to specifically attack the carboxy end. So they come at the carboxy end. So imagine here, I have this protein like this. Here's going to be, I'm going to make it linear like this. Here's an amino end on this side, and here's the carboxy end on this side. Let's say here's an amino acid, amino acid, amino acid, amino acid. These guys are like little Pac-Mans. And what they do is they come here on the carboxy side, and they just start cutting these at the carboxy end one at a time. So that's pretty cool. And by doing that, when they function like this, they're going to start releasing these amino acids out. So preferably, we'd like for them to spit out maybe some amino acids, an individual one, or they might even only be able to go to the point of 2 amino acids linked together, or maybe even 3 amino acids linked together. And again, you know what should all of these be? This should be a amino acid. This one right here should be a dipeptide. And this one right here can be a tripeptide. Okay? So cool, we know how these enzymes are functioning. Woo! Let's go to the next part. The next part is we get to the actual intestinal cell. This could be at different points of the small intestine. Primarily absorption of these different proteins is occurring in the small intestine, duodenum, jejunum, ileum. We're going to just not be too specific and just say this is a part of the small intestine and this is going to be an enterocyte, an intestinal cell. So let's denote that here, the top here. This is a enterocyte. Now, histologically, enterocytes, they do have, like if you look at an enterocyte here, they do have these cytoplasmic extensions called microvilli. These things right here are called microvilli. And these microvilli have a lot of these different types of brush border enzymes present on them. I'm going to represent it here in blue. Here's a brush border enzyme. And then I'm going to have another one which is going to be purple. This is going to be another brush border enzyme. The purpose of the microvilli is to increase the surface area for chemical digestion and for absorption. So what I have here on this surface is I have these things called brush border enzymes. And there's of two types. The purple ones, we're just going to say here, let's make it the aminopeptidases. We're going to make this the aminopeptidases. No preference here. I'm just picking whatever color just makes sense here. And then we're going to make the blue one the dipeptidases. Okay, so we're going to make the blue one the dipeptidases. Now, these enzymes are the easy ones. Again, they should be really easy, just like the carboxypeptidase. So here's the blue one. Okay, this is our dipeptidase enzyme here. This is one of our brush border enzymes. So it's dipeptidase. What do you think he's going to break down? Dipeptides. It's not that hard, right? That was one of the breakdown products. There was 3 breakdown products: dipeptides, tripeptides, and maybe even a little bit of amino acids. But what happens is this dipeptide here, let's use a different color here for these peptides in this case. I don't want to get it too confused here. Let's use this darker blue here. These dipeptides, let's put here, this is one amino acid, and then let's put this bond like this. Here is the other amino acid. That's my dipeptide. What the dipeptidase will do is, he's on that microvilli, he's going to bind onto these dipeptides, and he's going to break the peptide bond. When he breaks the peptide bond, he's going to release these into 2 individual amino acids. Okay? So these are going to be my amino acids. It's that simple. That one is a really easy one. Not too hard to digest on that one. No pun intended. All right? Next one is we're going to come down to this bottom guy. This guy is the aminopeptidase. Again, we've talked about this a lot already. So if we kind of have an idea of the chemical structure of these proteins, we know that a protein has to have a N-terminus, and on one end a C-terminus. That's one of the important things that you can see in proteins. Carboxypeptidases cleave at the C-terminus, the carboxy end. Aminopeptidases cleave at the amine end. So now, bring one of these. Let's keep it consistent here since we chose this color here. We'll put here some amino acids here, and let's put it like this. I'm going to put a tripeptide in this case. I could do a dipeptide, but I kind of want to do a tripeptide. And let's imagine here that on this end here is going to be the amino end, and on this end here it's going to be the carboxy end of this tripeptide. It'll cleave the bonds starting here at the amino end. So it'll go in order. Watch this. It'll go 1 and 2. So it'll cleave this bond first and then cleave that bond second. And again, the bond that it's cleaving is the peptide bonds. As a result, what is it going to release? It's going to release 3 amino acids. Oh my goodness, such a beautiful thing here. But here's the thing, it's not just amino acids. Sometimes it doesn't always, not all the tripeptides, not all the dipeptides get broken down, unfortunately, here in the actual lumen of the small intestine. Sometimes you might even have a little bit of some dipeptides here remaining. So here's going to be our dipeptide, and you might even have some tripeptides remaining, still here in the lumen. So this is our tripeptide. Okay, here's the cool thing. We have transporters for all of them. We have transporters for the amino acids, we have transporters for the tripeptide, and we have transporters for the dipeptides. Such a beautiful thing. Guess what that one's going to be? We're just going to keep them going in here in order. Dipeptide is going to go here. There's going to be a special transporter that will put this dipeptide into this enterocyte. And here, let's make room, so let's bring him up in the cell. Okay, so he's going to go up in this part of the cell. So there's the dipeptide. Same thing, tripeptide, this is going to be his little part. And the amino acids, they're going to funnel into this one. And we have one more mechanism that I'll talk about at the end, very, very briefly, because they're still trying to figure this mechanism out, what the significance of it is. Now, here's the thing. Dipeptides and tripeptides, when they get absorbed across the enterocyte, it's through a different mechanism than the amino acids. What's happening is, on this apical surface, there is a transporter. Let's do this one in pink. We haven't used this one in a little bit. There's these special transporters on the apical surface here, and what they're doing is they're pumping out into this area a lot of protons. And they're bringing into the cell some sodium ions. So this is a sodium hydrogen ion antiporter. These proton ions that we're pushing out here are going to get specifically used for the dipeptides, and it's also going to get specifically used for the tripeptides. So in order for the dipeptides and the tripeptides to get into the cell, they have to undergo a co-transport mechanism, a symport mechanism with the protons. So this is going to bring in the dipeptide. This is going to bring in the tripeptide. The last one is different. If you remember, we talked about this before. Let's do another pink here. Here's going to be another one. It's just a nice color. All right, and then what we're going to do here is this is on every cell in the entire body. Every cell. And what it's doing is it's constantly pushing 3 sodium ions out of the cell and 2 potassium ions into the cell. What is that doing? If you look at it, we're pushing sodium out. So what do you think is going to happen to the concentration of the sodium inside of the cell? The concentration of the sodium inside of the cell is going to decrease. But we're having a lot of sodium out here. So the sodium concentration, based upon whatever nutrients we're eating, my diet, I probably got high sodium all the time. But if you look here, the sodium concentration is probably going to be pretty high right here. So now if you look, we can go from areas of high concentration to areas of low concentration. That's passive diffusion. But the amino acids, they have to go from areas of low concentration to high concentration in the cell. So because of that, they're going against their concentration gradient. But if we bring sodium with the amino acids, it's going to help. It's the same mechanism for the protons. So now what I'm going to do is I'm going to bring these sodium ions into the cell. By bringing the sodium ions into the cell, it helps to be able to, by secondary active transport, bring the amino acids into this cell as well. Okay? Now here's the cool thing. Amino acids, I'm going to put AA here, my tripeptides TP, and my dipeptides DP. Unfortunately, when we put this into the actual blood, the hepatic portal vein, eventually it'll go into, we don't want them to be in tripeptides and dipeptides. So what we do here is we have special enzymes located inside of the cell. Let's do this in this orange color. We haven't used this one in a bit at all, actually. So dipeptides, tripeptides, they're going to have these different enzymes. We have different enzymes inside of the cell, and these enzymes are just peptidases. So this is going to be a peptidase, and this is going to be an intracellular peptidase. When these intracellular peptidases act on the dipeptide and the tripeptide, what do you think it's going to do? It's going to cleave those peptide bonds, giving us free amino acids. In this case, we'll have 3 here. In this case, we'll get 2 here. So now we have free amino acids. And then over here, amino acids. Oh man, this is so cool. Then we're going to make one big transporter. Let's use red here. One just gargantuous transporter. And this transporter, what it's going to do is, it's going to bring the actual amino acids out of the cell. It's going to bring the amino acids out of the cell and into this little area here where it can go into the capillaries, the intestinal capillaries, primarily like of the superior mesenteric veins. But then again, the superior mesenteric veins will combine with the inferior mesenteric veins, combine with the pancreatic veins, combine with the splenic, the gastric veins, and make this big sucker here called the hepatic portal vein. Again, we'll talk about that scheme, that circulatory scheme, when we do the liver lobule. Again, we do have another video on this where we talk about the thoraco-abdominal circulation, the veins, or a flow chart. Kind of gives an idea a little bit more about the hepatic portal vein. So go check that out if you want to know more about it. But what happens here is these amino acids, through this like facilitated diffusion mechanism, we can bring them out of these enterocytes. And then from here, guess where these amino acids can go? They can go into, and again, I'm denoting an AA here. These can go into the hepatic portal vein. From the hepatic portal vein, this will eventually take the blood to the liver. And then from this amino acids, we can use them for many things. You'll notice that the liver does a lot of protein synthesis. So he's going to convert these most likely into proteins. And there's also going to be a decent release of insulin during this feeding period, during the fed state, we say. So that's going to also help to be able to increase the protein synthesis and amino acid uptake into our cells. One last thing I want to talk about, and the only reason I want to talk about it is because they're believing that it's being related to certain types of diseases. One of those is they think it's related to celiac disease. We're not going to talk super in detail about this. So now, in different parts of the small intestine, primarily at the ileum, you have special types of cells called M cells, and you have enterocytes. What they have found is that we can actually absorb. Remember I told you that we generally don't want to absorb intact proteins? It's really hard. And the reason why is our immune system doesn't like to do that. We can do that in the postnatal period up to about six months for the passive immunity to be passed from the mother to the baby. But generally as an adult, we don't normally do that very much. But in 2 cells, enterocytes in the ileum, and these other ones called M cells, they can do what's called an endocytosis mechanism. Okay? They believe the M cells do through what's called clathrin-coated mediated endocytosis. But let's say here I endocytose a whole intact protein. So here's my whole intact protein. They believe that whenever we do this, the enterocytes can break them down by combining them with lysosomes and degrading that and releasing the free amino acids or free peptides afterwards into the blood, which can go to the hepatic portal system. But the M cells do something else that's really interesting. They found that the M cells can take and actually not break this protein down. They don't actually break it down because they don't have a lot of lysosomes. So they keep the protein actually intact. And when they keep the protein intact, they actually can release this protein out on the basolateral surface. So imagine here I release this protein out into the basolateral membrane. So now here I'm going to have this protein released onto the basolateral membrane. When it does this, there's a connective tissue right underneath the epithelial cells, and this connective tissue is usually areolar connective tissue. They call it the lamina propria. Well, what happens is we have special types of cells, immune system cells out here. These immune system cells are called macrophages. There's a lot of those. And what these macrophages do is they naturally are tasting and controlling what's leaving, coming from the GI tract into the blood. Well, what this macrophage will do is they believe that the macrophage can respond to this chemical, it can phagocytose it, and present that actual peptide on the cell membrane on what's called an MHC2 complex. And this peptide, after it's expressed here, it can take it to different types of immune system cells. They believe it can take it to what's called T cells and trigger an immune response by the release of different types of cytokines. Now, why is that important? They believe that this plays a role, the M cells play a role within what's called mucosal immunity. But at the same time, they believe it could be related to certain diseases because there is a protein in gluten called gliadin. And whenever gliadin gets released over here in certain people, their macrophages will attack the gliadin, express it as MHC2 complexes, take it to the T cell, and the T cell will start releasing tons of different types of chemicals that trigger a massive inflammatory response. So if it did that and it brought it to a T cell, and it did this reaction here where it presented it to the T cell through what's called CD4 proteins and a TCR, it could trigger this T cell to start releasing chemicals like tumor necrotic factor alpha and gamma interferons, which trigger a massive inflammatory response and can lead to a lot of problems in people with celiac disease. All right, Nation, so I hope you guys enjoyed this video on protein digestion and absorption. I hope it made sense. If it did, please hit that like button, comment down in the comment section, and please subscribe. Also, if you guys get a chance, go check out our Facebook, our Instagram, maybe even our Patreon account. All right, Ninja Nerds, as always, until next time.